I've had the privilege to do research with him for the last 3 years. And what I'll be presenting on today is entitled Lagging Behind, Understanding the Importance of Circadian Rhythms on Inflammatory Pathways. And just to give you a little bit of a heads up how my presentation is going to go, I'm going to give you some background information to understand a few things that are going to be stressed throughout my presentation. And then I'm going to go over a research paper that inspired our work within the lab, as well as my experimental results themselves. So here we go. Okay, so first off, I'd like you to understand the importance of sleep, especially the circadian rhythms, and why sleep is essential. According to the National Sleep Foundation, our body runs on a 24-hour endogenous clock. And they recommend that we get about 7 to 9 hours of sleep a night. Now, why is getting so much sleep that important to us as humans? Well, sleep is in charge of recharging your brain. It's in charge of releasing important hormones within your body, as well as consolidating all of the facts that you take in throughout the day, everything that you're learning. And it repairs your cells. So when you have a weakened immune system, your circadian rhythms are responsible for helping your cells get better. So it controls how often you get sick, but not only does it control how often you get sick, it too often controls how much fat you have within your body, because it's a big regulator of your metabolism. So what you eat affects your body as well. So now that we understand that sleep is very important to us, what exactly is going on physiologically within these 12 hours during the day and 12 hours at night? Well, if you're indeed getting that 7 to 9 hours of sleep that I hope all of you are getting, you're probably entering your deepest sleep cycle around 2 o'clock in the morning. From there, at 6:45 in the morning, you should have your sharpest rise in blood pressure, and this is normally the time that many adults will be waking up in the morning. From there, right about now, you guys are probably experiencing your fastest reaction time, and it's not uncommon for you to see sports practices at this time because of that fact, that you're probably able to react faster to that ball coming your way, or that pitch coming your way in the batter's box. So at 6:30 p.m. tonight, you're probably going to experience your highest blood pressure, maybe from something stressful that occurred throughout your day. And at 10 o'clock p.m., your working ability decreases entirely. And this is probably when you're most likely to be just dragged down from the day and wanting to just fall asleep. So now that we understand these physiological processes, what happens when you interrupt this 24-hour cycle? Well, some implications of sleep deprivation throughout the body include decreased accuracy, as well as tremors and body aches, irritability, cognitive impairment, hallucinations, as well as lapses in your memory, an increase in the risk of obesity, as well as heart disease, causing for an increase in your heart rate, as well as an increased risk of type 2 diabetes. But what the deprivation that I am most, or the implication that I am most interested in involves the impaired immune system. So that's what my research is essentially going to be based on. So now what happens, how does our body get deprived of this sleep? What are common causes of this sleep deprivation? Well, being a college student, as many of you should know, you don't get very much sleep. In fact, this is me in the lab running one of my time trials at midnight. Homework causes lack of sleep, working on talks and presentations like this that you have to give today, as well as world travel and jet lag. Now, what exactly is jet lag? Well, jet lag involves traveling abruptly from country to country, as well as, anywhere across, coast to coast in the United States. Often see people in airports like this whose jobs bring them to travel quite frequently, just exhausted. Rotating shift work, people who are seen to work third shift, as well as the cemetery graveyard shift, as they call it, are seen to experience repeated exposure and increased risk of lifestyle-related diseases. And that's really interesting because as of late, they've been discovering that nurses who work third shift have seen to have an increase in breast cancer, as well as obesity and type 2 diabetes. So jet lag can really be detrimental to your circadian rhythm. And what disrupts your jet lag consists of light and dark cycles. So now that we understand a little bit more about jet lag, we're going to get into how the circadian rhythm is entrained, or how that 24-hour cycle is prone to know what time it is. Well, like we just mentioned, light is a huge factor in that, because when your body wakes up in the morning, it sees blue light. That alerts it that it needs to be active and ready to go. And at night, when your body shuts down from this light, it thinks, okay, it's time to go to bed. But how many of you are on your cell phones before you go to bed, checking those Twitter updates and things like that? That blue light from your electronic devices is interrupting your circadian rhythms and causing you to be more alert at night. So it's harder to fall asleep at night if you're on your electronic devices due to that blue light. Now, food is a major contributor to the entrainment cycle of your circadian rhythms as well, because your body depends upon glucose for its metabolism. Glucose tells you, if you have low glucose levels, it tells you your body is low on energy and it needs food. So your circadian rhythm controls when you need to eat and your amount of energy that you're going to have. So if you're not putting the right substances in your body's not going to be working to its fullest potential, and its circadian rhythms are going to be disrupted. Now, right here in the brain, we have something that's called the suprachiasmatic nucleus, and that's going to be referred to as the SCN from here on. The SCN is located in the brain, and it's responsible for the release of numerous different chemicals that synchronize all the cells within the body. The body has 3 trillion cells synchronized by this one tiny portion of your brain. And it does so by the release of these chemicals into your bloodstream. Now, scientists don't exactly know how this happens, or the full scientific reason as to how your body can control 3 trillion cells like that. So, given this information, I thought it would be really awesome to look at one of those chemicals released by your suprachiasmatic nucleus pertaining to vasopressin. Vasopressin really intrigued me because it's a factor in a bleeding disorder that I have. So I thought, what better than to study vasopressin and the circadian rhythm, knowing that this affects my bleeding disorder and I don't get very much sleep at night. So I found a paper entitled "Mice Genetically Deficient in Vasopressin V1A and V1B receptors are resistant to jet lag." And delving into this research paper, to give you a little bit further information about arginine vasopressin, it's a gene that encodes brain peptides, and it has 2 different receptors, as I just mentioned. And one is called V1 receptor, and the second is the V2 receptor. And both are synthesized and released by the SCN. The V1 receptor increases your systemic vascular resistance, and the V2 receptor increases your blood volume. Both of them having control in regulating your blood pressure. Now that we understand this, I'm going to delve into the hypothesis of the paper, which stated vasopressin is important in the re-entrainment of the body. They proposed that since vasopressin is released by the SCN, that it might have some contributions to the circadian rhythms. Now, what exactly is this re-entrainment that I'm talking about? Well, right now it's about 3:20 right here in the United States. Say we jump across the ocean and are in London right now. It's about 10:20 there. So just because we know it's 3:20 here in America, our body doesn't know that there's a time difference when we go to London. So it's much harder for our body to adjust to these different light cycles. It takes 1 hour per day for your body to re-entrain when it's introduced to a different time zone like this. So it would take 7 days if we were in London right now to know that our body is able to readjust its circadian rhythms to that of London's time as opposed to here in America. So delving into the methods of the paper, they specifically took controlled mice and exposed them to jet lag conditions. And they measured how much jet lag condition was being displaced upon the mouse due to spontaneous locomotor activity measured by infrared sensory systems. And then they monitored the mouse's activity by the standard wheel running. Now that we understand the experimental side of it, we're getting a little bit further into the pharmacological effects of this experiment. So what they did essentially, once again the SCN here, they applied antagonists over the SCN via a mini osmotic pump. And these antagonists were responsible for blocking vasopressin, both the V1 and the V2 receptors. This blocking of the receptors, corresponding with exposing the mice to separate light and dark cycles over an 8 to 12 hour period, led to the following results. On the y-axis here we have our time and days, and on the x-axis here we have a control mouse, a mouse exposed to a smaller amount of the V1 antagonist, and then a mouse with a much larger amount of vasopressin antagonist. So what this diagram shows is that vasopressin antagonists enhance the speed of reentrainment of mice subjected to jet lag much faster than a mouse who didn't have this vasopressin blocked. So the larger the antagonist applied, the sooner the mice could regulate its circadian rhythms back to normal after being exposed to jet lag. So what does this all mean? In a nutshell, mice lacking vasopressin V1 and V2 receptors are indeed resistant to jet lag, but just because a mouse might not be lacking those receptors doesn't mean that there could still be an aid in helping to fix the jet lag the mouse would be encountering. Because if you block those receptors with those antagonists, that'll help in the jet lag as well. This led to us kind of thinking that vasopressin would be a possible therapeutic result in humans, and that interested me greatly because I thought, well, what about humans and their circadian rhythms? Is there an experiment that we could do with that might help us fix jet lag conditions for when humans are traveling or working that third shift? But Dr. Dave and I realized it's very hard to make a knockout human. We can't just chop your leg off and fix your entrainment cycles in there like we could to a mouse. But so we had to figure out some way to obtain human cells. And we realized that the fastest way and the easiest way for us to do that would be by looking at blood cells. Because blood cells are already entrained and are cycling outside the body still. So what sort of experiments could we do with that blood? Well, we did immunological experiments. This led me to a second question, thinking, well, why do we feel so awful at night when we're sick? In order to understand why we feel so bad at night, we first need to look at the immune system. And our body has two different types of immune systems. It has an adaptive immune system as well as an innate immune system. Due to our time restraints, I'm only going to be talking about the innate immune system. The innate immune system is extremely important to your body because it's immediately recognizing the influence of pathogens. When something's in your body that shouldn't be there, this is the first system to react. And it recognizes pathogens because of the pathogens have a specific pattern. And it does so by the use of macrophages. Macrophages are sentinel importance to the immune system as they are the first ones, whether it be in the daytime or nighttime, to recognize these patterns of the pathogens. And in doing so, once it recognizes that something's in the body that shouldn't be there, it automatically releases inflammatory mediators in response to that stress. And those can be pro-inflammatory mediators or anti-inflammatory mediators known as cytokines. So, given all this background research, it led me to hypothesize that macrophages respond to pathogens based on your circadian rhythms. And knowing this, I would expect there to be a greater inflammatory response at night than in the daytime. So what exactly is the cytokine that I was just talking about? Well, the cytokine is an inflammatory mediator. It's very important because it's a protein responsible for cell signaling when there's a stressor in the immune system. So it's produced by these immune cells like macrophages, and it's released in response to the signal transduction pathway, which is triggered by pathogens. I'll be getting into the signal transduction pathway in just a minute, but first I want to explain the pathogen that I've introduced into my cells. And that pathogen is known as LPS. LPS is a lipid polysaccharide. It's a molecular pattern that those macrophages recognize right away. It's part of the cell wall of gram-negative bacteria. And it binds macrophages, promoting the secretion of pro-inflammatory cytokines. So whenever there's a stressor, the macrophages are the first to know based on this pattern in our experiment. So what exactly is the signal transduction pathway? It's basically a large game of telephone. It's telling your body, based on what's going on inside the cell, it's a connection to the outside of the cell, saying there's a stressor in our environment, we need to do something about this. So when the LPS comes in here, it binds to our receptor and elicits a numerous amount of conformational chemical changes. And these chemical changes are known as transduction. And what that is doing right there is activating the phosphorylation of this protein, the P38 kinase. So what is this P38 kinase? Well, the P38 kinase is a very important protein that responds to stress and stress stimuli. It results in the release of many of these pro-inflammatory cytokines, and the phosphorylation of that can once again be seen all over in here, leading to a series of different reactions. After P38 is phosphorylated, our next chain message being sent down the telephone line is in the NF-kappa B pathway right here. NF-kappa B is held in the cytoplasm by I-kappa B alpha. Now, what are NF-kappa B and I-kappa B alpha? Well, NF-kappa B is a very important gene involved in inflammation. Or, I'm sorry, it's a protein that controls many genes involved in inflammation. And I-kappa B is responsible for holding NF-kappa B within the cytoplasm until that stressor occurs. And when a stress occurs, interrupting that NF-kappa B pathway, then I-kappa B is degraded. And NF-kappa B goes directly into the nucleus and stimulates transcription of those inflammatory mediators. And one of those inflammatory mediators that's going to be transcribed is MCP1. MCP1 is a representative inflammatory cytokine known as macrophage chemotactic protein. It's very important in regards to recruiting inflammatory cells to the sites of injury or infection. And that can be seen happening right here when mRNA transcribes for this cytokine, MCP1, going on in the nucleus right here. So now that we know all this background information and how your cells are communicating within your body via your signal transduction pathway, we need to take a further, deeper look into regards to how the circadian rhythm feedback loop is really working. So our circadian feedback rhythm loop here is a 24-hour endogenous cycle controlled both by light and the food that you eat. Its transcription pathway regulated specifically by this clock protein. Now, all of these are transcription factors. Some are more prevalent during the day, and some are more prevalent during the night. Now, what happens if you oppose this expression feedback loop? That might challenge your body in numerous different ways, leading to an offset in your entrainment cycle, setting you up for human disease, and particularly those diseases could be anywhere from obesity to diabetes or heart disease, like we just mentioned. Now, even though I would love to delve into all of these transcription factors, I only have time to really choose one, so I chose to focus on the RevRB. RevRB is a circadian regulated gene, and it contributes to our energy metabolism. It's a day transcription factor, so we'd expect to see more of this transcription factor during the daytime hours. And it's important because it helps us link the circadian rhythms with the signal transduction pathway. All of that background information is now going to help you understand what we had to do for my experiment. So our first mission was to obtain worthy patients so that I could obtain some blood samples. So Dr. Dave was very generous and let me stab him with a needle numerous times to get blood samples. And also some Bio 130 students working on my project with me were very influential in helping me get blood samples from our stockroom supply manager, as well as some of them graciously donating to my experiment as well. So once we had these blood cells, we added a sugar solution to the blood in hopes to separate out the red blood cells from the white blood cells and the remaining serum based upon density. Since white blood cells are less dense than red blood cells, we were able to harvest the white blood cells, taking the white blood cells for our study, as we wanted to focus on macrophages. Now, once we had those blood cells, we did a time trial, and the time trial was done both at noon and at midnight. So this is a picture of our time trial. We had our control here, and every 15 minutes we added LPS, which once again was that lipid polysaccharide that I talked about earlier. And we added LPS every 15 minutes until we reached an hour. After the LPS lysed our cells, we incubated the cells and isolated our specific protein of interest, being the I-kappa B alpha as well as that P38. And then after the time trials, I did a SDS page gel as well as a Western blot. Now, what is an SDS page gel and a Western blot? Well, STS page gel is this little picture right here, and essentially consists of gel electrophoresis, in which I'm separating my protein out by size and charge. So at the top here, we're going to obtain our proteins that have the largest charge and the largest size. And then down here, we're going to have the smaller proteins resulting. Now once those P38 proteins are isolated, I transfer those via a Western blot to another kind of gel-like membrane. And then I block that membrane, and I incubate it with primary antibodies of interest. Once that happens, I go through a series of washes and add another secondary antibody so that I can just wipe out everything except for the one antibody that I wanted to be looking for, which indeed was the NF-kappa B. I wash it one more time, and then I image the blot so that I can see the protein expression and analyze that further on. So now getting into the results of that image. In regards to a normal healthy individual, this graph is showing our P38 phosphorylation. So right here we have our daytime trial for these two pictures, and right here we have our nighttime trial. And here is showing our time in minutes from 0 to 60 minutes. And we're looking once again at this protein kinase P38. GRB2 here serves just as a loading control to make sure that the same amount of protein was exhibited throughout the gel. So what is the difference between daytime and nighttime here? Overall, this shows that P38 was degraded much more at night than it was during the day. And this is extremely important because it shows that there is a difference between P38 protein levels at daytime and nighttime, showing that there might be an effect from an inflammatory mediator during the day and the night, as we get into our I-kappa-B alpha degradation. Once again, in a normal healthy individual, given the same day and nighttime trials with the 0 to 60 minutes, we looked at I-kappa-B alpha degradation. And corresponding to our night, a greater amount of I-kappa-B alpha was degraded during the day than was at night. So showing that, once again, there was a difference between daytime and nighttime in regards to the expression of this protein. Now that we looked at a healthy person, we wanted to take into consideration a sick person. A sick person had a greater degradation of I-kappa-B alpha at night than it did during the daytime. Now this is the exact opposite of a healthy person. Although not as much was degraded during the day, what we need to focus on here is that there was essentially a difference at nighttime in which more I-kappa-B alpha was degraded at night than during the day. So a summary of these results. A healthy person had a greater phosphorylation of P38 during the day, which matched up with our I-kappa-B alpha data showing that more inflammation during the day due to the degradation of I-kappa-B alpha, and then likewise confirming that there was indeed a day-night difference between I-kappa-B alpha. In a sick person, there is less degradation overall, showing that less inflammatory cytokines were released and that the immune system was much weaker in a sick person than in a healthy person. There's a low variability of degradation between day and night for a sick person because those macrophages are exhausted. And you're not likely to be moving around as much when you're sick. So your circadian rhythm is going to be altered in that regard too because your activity level is going to be decreased. So there's slightly more inflammation at night, but that was not sustained. Given all of these conclusions, we now need to confirm two things. Are the cells really in a rhythm? Are they really working in this entrainment? And then are inflammatory mediators indeed being made? Well, how are we going to test that? We're going to test this via qPCR. And qPCR is known as quantitative polymerase chain reactions. So how does qPCR work? Well, first we have to isolate our RNA from our samples. From there, we're going to be turning that RNA into cDNA. And once the cDNA is made, we're going to do a series of different quantitative polymerase chain reactions to obtain mRNA. And once we measure those mRNA levels, we figured that we could obtain how much protein was being expressed because mRNA gets translated into proteins. So now we have these very confusing graphs. And don't worry, I'm going to sum it up for you and make it extremely easy to understand. This is our qPCR data for a healthy individual as well as a sick individual. And basically what we have going on the x-axis here is the number of cycles being done, and then our fluorescence measurement here. And we're specifically looking at the mRNA expression of MCP1 as well as RevRB. And what is expressed in our healthy individual, in that there's a fewer amount of cycles for the MCP1 and RevRB being expressed, indicating that there's a larger concentration of those genes during the day than there is at night. As you can tell, that it takes many more cycles to occur over here for that MCP1 and RevRB to be expressed, indicating there's a much lower concentration of those genes. So in summary, our qPCR expressions showed macrophages do experience circadian rhythms in vitro, and that was shown by our RevRB being a larger concentration during the day. And there was an inhibition of the P38 kinase, therefore resulting in increased MCP1 and RevRB during the daytime as opposed to the nighttime. And ultimately, our results were inconclusive as to whether or not MCP1 expression was altered between the day and night after exposing macrophages to the LPS. So that's something we want to take into further consideration for our future. So in conclusion, a healthy person shows a greater inflammatory response during the day, which was the opposite of what I thought would happen. And that's mostly because the cytokines are functioning in the immune system much better when you're active and when you're able to keep your body and the circadian rhythms going, as opposed to when you're feeling sick and your circadian rhythms aren't getting as much activity as they need, or really what they're used to because your macrophages are exhausted. So a sick person has lower inflammatory response than a healthy person because your macrophages are exhausted, like I just said. And that sick person has little change in their inflammatory response during the day than at night, and that's likely due to our reduced activity. In regards to future directions to my study, I would definitely like to utilize more blood samples and leave Dr. Dave's veins alone for a while. But not only would I like to do that, I would like to use different inflammatory mediators other than the MCP1, and I would further like to analyze the release of MCP1 because we were only able to look at that via qPCR and mRNA expressions from that, and those were inconclusive. So in regards to acknowledgments, I would really like to thank Dr. Dave because he's been a huge influence on my life here at Lawrence and my research especially. This is my predecessor in the lab, Brielle Fleming, and she was very instrumental in helping me get down all the techniques I needed to know to be able to do SES page up and Western blots, and as well as my Bio 130 friends who helped me extensively with the numerous time trials so that I could get some sleep at night for once. So these are the references in regards to my presentation, and if you have any questions, I'd be happy to answer them for you. Great.
I had a question about how you treated the blood samples, because if you take the blood out of the person's body, do you have to set a light with a timer to keep the blood circadian rhythm normal?
Everything was done immediately after we took the blood samples. We automatically centrifuged them after applying a sugar solution so that we could harvest them right away and get the incubator going. And once we had the cells in the incubator, then we were able to do time trials at noon and at night. So that light wouldn't affect them. Dan?
So then you drew all the blood during the day, you never had Dave come in at 10 p.m. to get ready for a sample,
No, I don't think Dave would be
too pleased. Right.
pleased to do that.
So then how does, I think what Rich was asking is, how do you account for that waiting time? If you wait, say a day or two and you leave them in the incubator, how do you make sure the cells don't get thrown off? Because if they're not receiving signal from the body anymore.
I think what was really crucial in that regard is we never waited. Anytime we took the blood, we automatically put them in the incubator and ran the noon trial and the nighttime trial within that same day.
Okay.
So that wasn't really an effect on our experiment.
Okay, cool.
Yes?
Do you do you anticipate seeing any differences if this study were done not to humans but to some sort of nocturnal animal where they have a different type of circadian rhythm possibly?
I think that would be definitely very interesting to study. I haven't really looked into that in regards to
I'm just curious.
nocturnal, but I don't know. That would be fascinating, especially because we thought inflammatory mediators would have an effect on why you feel worse at night. So I think it would depend upon the release of cytokines within that species and overall effects in that regard. Because ours was showing essentially a focus on those macrophages and the pro and anti-inflammatory cytokine release from there. So I think it would depend on how we harvested cells from them and what their entrainment would be. Dan?
Would you anticipate any seasonal variation? For instance, testing patients in the summer when the day is much longer, as opposed to the winter when the night is a whole lot longer?
I think that's definitely a feasible possibility because it depends on your light and dark cycles, and you know, once daylight saving time comes around, our body is definitely extremely affected by that 1 hour of sleep we either gain or miss. So I think that could definitely be very plausible. Melissa?
Did you use your blood samples at all, or with your blood condition? Did that affect anything? Did you use that?
Dr. Dave and I were not sure if my blood would be able to be tested because it does have, I guess, a concern in regards to that vasopressin. So we thought I should avoid being tested on because we didn't want that to add any adverse effects to our experiment. Jackson?
Would you expect any differences in the regards of the adaptive immune system? I know that you worked with the innate immune system. What would you kind of expect from that?
Definitely. So the innate immune system recognizes patterns from pathogens within 0 to 6 hours in the body, whereas it takes the adaptive immune system 2 weeks to recognize those pathogens. So I think a lot of bad things would be occurring within your body by the time you would be able to know it if you were looking at the adaptive immune system. So ultimately the innate immune system I think gives you the best focal point because within 2 weeks time a lot can happen damaging those genes in your body if a pathogen interrupts those pathways, especially the NCAP-B pathway.
Okay. Thank our speaker again. Okay, 5-minute bathroom break and then we'll have Bennett.
Thank you everyone for coming.